Anyway, okay, so yeah, so this is the lecture for today, pathogens from the past. Now, our previous lecture was called Waking the Dead, and we looked at ancient DNA, and there have been many spectacular successes using ancient DNA to study human evolution, the Neanderthals, the Denisovans, things we didn't mention. There's been genome sequences from Ötzi, this ice mummy from the Alps, Paleo-Eskimo, and so forth. But one of the problems you get when you're doing ancient human DNA studies is that the DNA of the handler can contaminate your samples. And this creates a big problem in trying to distinguish between the contamination that's coming from modern genomes and that's actually originally there in the sample. Another problem back in the days when people used to do PCRs was that it's quite possible for your laboratory environment to get contaminated with your PCR products from one round of PCR, and they can find their way into the next round. Now, if we look at ancient pathogen DNA, there's now substantial literature on this, and I'm going to talk to you about leprosy, plague, and TB, but there are others as well. Here, of course, the risk of contamination, biological contamination, if you like, is much lower because we're assuming that the people handling the specimens, extracting DNA, making libraries, do not have plague, do not have TB, do not have leprosy, so they're not going to be putting those organisms into the samples and into your DNA and so forth. But nonetheless, there is still a great deal of skepticism in the field, particularly when people used to rely, and there's a substantial literature where people use PCR. And the problem with PCR is, as I mentioned, that you get this amplicon carryover, and that remains a problem if you're doing that kind of approach with ancient pathogen DNA. So we've moved on to looking now not just at PCR products but at whole genomes, and I'm going to give you 3 case studies here, starting with Yersinia pestis, causative agent of plague and the Black Death. Then we'll look at Mycobacterium leprae and the causative agent of leprosy, and then Mycobacterium tuberculosis and its role in TB. Now, one thing I ought to say is that we don't just study these ancient genomes from these pathogens to understand how the pathogen has evolved. We also integrate that into our knowledge of the population genetics of current pathogens. So if you remember when we were looking at human DNA, we said, right, we look at the Neanderthals and we can look at all modern variation in modern humans and we can fit that in. We can do exactly the same kind of thing when we're dealing with bacterial pathogens. We can look at what do we know about the genomes of the organism that causes disease today and how much variation there is across the world and what are the various components. We can look at all that and interpret the ancient DNA studies in the light of those studies. So let's start with Yersinia pestis, which is the causative agent of plague. Now, this generally is not spread from humans to humans. It is possible to spread from humans to humans, but usually there's an animal reservoir out there and it spills over into human populations from that animal reservoir. And there are a whole range of rodents. I mean, rats are one particular reservoir that can where we can catch it, but there are others as well in different parts of the world. But it spreads from those animals not directly, but typically through flea bites. And what you end up with is a picture like this, where you've got bubonic plague, and the term bubonic plague comes from these things here called buboes, which are enlarged lymph nodes, which in the worst cases they actually grow so large they erupt out of the skin. And so here's an example here, these inguinal lymph nodes. There are other varieties of plague. In addition to bubonic plague, it can present in the septicemic phase, where you get bleeding and necrosis. Here's a leg where you've got widespread ischemia and necrosis in the foot there. Presumably have to be amputated. And then the worst kind of plague in terms of mortality rates and transmissibility is pneumonic plague. When this organism gets into the lungs of a patient, they can be coughing up very large amounts and aerosolizing very large numbers of the bacterium, and they present a risk to anyone they come into contact with who can then catch that as well. So this just summarizes what I've said in diagrammatic form here. The flea is at the heart of this. Humans infected because a rat is carrying plague bacteria. The rat is bitten by a flea, which drinks the rat's blood. The bacteria then multiply in the flea's abdomen, and the flea bites humans and infects humans. And what we know from looking at the biology of the pathogen, looking at genomes, and what we can look at the relationships with other organisms, is that we know that this organism, Yersinia pestis, sits within this genus Yersinia, and there are two other kinds of Yersinia, Yersinia interpelicus and pseudotuberculosis, that can also cause infection in humans, but they usually cause infection via the gastrointestinal tract. And somehow Yersinia pestis has evolved to cause disease through this additional route, this route of the flea biting individuals, infection spreading to the lymph node and then going on through the blood to then be transmitted to cause widespread infection in humans. And so it's an interesting kind of sideline that this organism has developed, a particular niche that it's developed to cause disease, that differentiates it from the rest of them. And the reason we're interested in Yersinia pestis and plague is that this condition has played a key role in human history, as it's been responsible for huge numbers of deaths. So in the 6th century, we had the Justinianic plague. At its height, there were recordings of tens of thousands of deaths per day. And it is said, I don't know how much truth there is in this, that the effects of this plague were so substantial on the Roman Empire, particularly the eastern surviving part of the Roman Empire, the Byzantine Empire, and on the Persian Empire, that when the Arabs erupted with the rise of Islam out of the Arabian Peninsula, those pre-existing empires were no longer able to mount a substantial military response. And so you could argue that the plague has played a role there in the movement of peoples and ideas. Then the Black Death is the name given to an episode of the plague in the European population in the 14th century, and it's said that around a third of the population died from plague, from Black Death. And this led to substantial depopulation of the various countries involved, meant that there was a loss of labour in the rural areas. It's said that this brought about the end of the feudal system, because labour was in such short supply that the lords of the manor couldn't just order people around anymore, because there were so few of them and they could just move to the next place. And it also is said that this may have changed the structure of Europe to pave the way for the Renaissance. Then we had the Great Plague of London in 1665, and then from the 1860s onward we have this third pandemic. So we described the Justinianic plague as the first pandemic, the Black Death as the second pandemic, and now the third pandemic. And we can see the effects of plague in history when we look at these kind of wood cuttings of skeletons dancing about, and these illustrations of people in their beds covered in buloes and spots, and these plague pits. So when any major building work goes on in London, they have to get the archaeologists in first. The archaeologists often discover these kind of plague pits where individuals have died rapidly. There's no chance for a normal Christian burial. They just chuck them all in a big pit together and bury them that way. So this animation here shows you the spread of the Black Death through Europe from the east, from starting off in the Byzantine Empire and moving across what was the Ottoman Empire back then. The thing is that plague has not gone away. It is still out there in animal reservoirs around the country. When I first gave this lecture a couple of years ago, there was a large outbreak in Madagascar underway. There were over 2,000 cases, and there were over 200 deaths from that particular outbreak. And just last week, looking at the news headlines, there has been some cases of plague described in China just in the last few weeks. 3 cases. Fortunately, they're not epidemiologically related. This does not appear to be the start of some large outbreak in China. We do see sporadic infection with plague in various places around the world every now and then. It's only in the rare cases that it then spreads out to becoming an epidemic. So when we looked at genomes, one of the things we discovered was that actually Yersinia pestis is a funny little thing. It's actually a clone of Yersinia pseudotuberculosis. And so it's a variant, it's a species kind of buried within a species, if you like, where it evolved from this much more diverse species, Yersinia pseudotuberculosis. And what we've been able to piece together from the genome sequences is that there was a transition from Yersinia pseudotuberculosis as primarily an enteric pathogen, causing infection of the lymph nodes associated with the gastrointestinal tract, to becoming a flea-borne pathogen. And there were various stages that needed to be gone through to make this happen. So the first was the acquisition of various plasmids that encode virulence factors that are needed for plague to work as plague. So this plasminogen activator allowed the spread in the bloodstream more quickly from the intradermal site of infection. A protein capsular antigen and the so-called murine toxin were needed for survival in the flea. In addition to that, there were a variety of pathogenicity islands, as we call them, which is a regions of gene clusters that have been inserted into the chromosome of the pathogen. And also, the third stage of this was that many of the genes that were needed for Yersinia pseudotuberculosis to live in its more generalist lifestyle were lost. And we actually, when we look at the Yersinia pestis genome, we see lots of broken genes, what is often called pseudogenes, lurking in there. And the genome is undergoing this process of degradation. So here's a paper describing that from a former colleague of mine, Mark Uckman, back in the late 1990s, showing that Yersinia pestis is a recently emerged clone of Yersinia pseudotuberculosis. But one of the key questions that arose in this field was that, well, okay, we've got Yersinia pestis today, and we call that the agent that causes plague today. But how do we know that the Black Death, this historical event that's recorded in the literature, we can find evidence from archaeology in the plague pit, how do we actually know that that's the same disease? Could it be that we've confused things over time? And so here's an article that came out in 2001. So the disease that spread like wildfire is still the most violent epidemic in world history, killed a third of the population. As far as most people are concerned, the Black Death was caused by bubonic plague. But 2 epidemiologists from Liverpool say we've got it all wrong. In the book they say that if we look at the way that plague spreads today, and we look at outbreaks, and we look at the historical records, there seems to be a disconnect. It doesn't seem to be behaving the same way. What was it that could account for the fact that it spread like wildfire through Europe, when today it's, occasional tip-overs from rats? Why would it suddenly take off like that? And so there was some skepticism as to whether we could roll these two things in together and just say, yeah, plague back then in the historical narrative and plague today are the same thing. So the first efforts to try and work out an answer to that question came from PCR approaches, and one particular guy, Didier Raoul, a rather maverick microbiologist based in Marseille, actually probably one of the most influential microbiologists of his generation. He said, well, because what people were saying was, oh yeah, you can do a PCR and you can get some signal out of a sample, but it's because you've got contamination in your lab. So what he said was, well, what we'll do is we'll do what we call a suicide PCR. We'll make some primers that are targeted towards Yersinia pestis that we've never used before in the lab. We'll just use them once to analyse one set of samples, and then we won't use them again. And so the fact that we get a signal the very first time we use these primers, we've never done PCR with them before in the lab, means it can't be carried over from some previous experiment, from your positive control or whatever. It has to be real. And so he was one of the first to say that, yeah, they've got these, this is some teeth of a child and 2 adults from 14th century grave in France, and they did this suicide thing, and they showed that a positive result followed by sequencing, and negative controls were negative, and they basically showed that we can end the controversy that medieval Black Death was plague. Now, around the same time, I was working with this guy, Mike Prentice, who was when I was at Barts, and he said, "Don't believe these French people. You know, if you can't trust what they're saying. Didier Raoul is just, you know, he's got a large mouth and speaks loudly, but it's all dubious." I said to Mike, I said, Mike, he had got access to plague pits from via the Museum of London, and I said to him, "You're on a hiding to nothing here, because if you try and replicate his results and you get no results, you get negative results, that's a negative finding. Generally you can't publish negative findings. Whereas if you do get positive results, then everyone will say you've got contamination, just like you're saying he's got contamination. So you can't win with this." But he ignored me, and he did go on, and he managed to write up his work to show that they could not get any amplicons from these plague pits. And so the controversy continued. This was 2004, for a bit longer. Fast forward a few more years though, and people doing ancient DNA studies got slicker and better at what they did, and we finally got, from Barbara Bramante, Elizabeth Carniel, Stephanie Hench, and various others, we got this first genome sequences of Yersinia pestis. They showed that basically, sorry, I'm getting ahead of myself here. They showed that they could get signals from these plague pits, and they found by amplifying things and looking at SNPs that they could actually start to make inferences on the basis of that. Sorry, I got ahead of myself there. It was a little bit later, the next year, that we got the first genome sequence from Yersinia pestis from victims of the Black Death. And here you can see the last one here is Johannes Krause, who has risen up to become the biggest name, basically, in the field of ancient DNA applied to pathogens. He also does extraordinary work on ancient human DNA as well. And here he's basically saying we've got technological advances in DNA recovery and sequencing, and they reconstruct the ancient genome of Yersinia pestis at 30-fold coverage from Black Death victims, securely dated to episodes of pestilence-associated mortality in London. And so what they showed there was that they got these genomes, this genome that came out of the Black Death. It clearly was very closely related to the modern Yersinia pestis genomes. But when they drew a phylogenetic tree, what they were able to show was that their genome sat at the base
of the tree based on all the modern sequences that are available. We've got all these sequences here available from Yersinia pestis, various branches and so forth. What they showed was that their Black Death genome sat at the base of that phylogeny, and that's what you'd expect, because when you reconstruct all the mutations that have given rise to variation here, and you go back to that node there, you'd expect that the ancestor of all the modern strains to look like the Black Death. So it kind of made perfect sense in terms of it being the Black Death genome. And you can see there in pink the Black Death, the Justinianic plague quite substantially earlier, and can't really say anything about that at this stage. And I have to say, at the stage that this was done, it was one of those miracles that knocked you off your chair. Wow, they've got a Black Death genome. Never believed that they would ever get earlier genomes than that, say from the Justinianic plague. But again, the subject moves fast forward, and a short while later they did get some individuals there from this Justinianic plague, and they isolated DNA from the teeth. Teeth have become actually, in this field, a very prized specimen, because if you've got unerupted teeth in particular, but even erupted teeth, when the person dies, they get kind of sealed off from the rest of the circulation, and the pulp in the teeth is kind of like a time capsule sampling the blood that was in the patient at the time of death. And they reconstructed these various genomes, did radiocarbon dating, and they found that that they got a novel branch that had no contemporary representatives. So it was either extinct or hadn't yet been sampled, but it does interleave between two extinct groups and is distant from the strains associated with the second and third pandemics, the Black Death pandemic. So again, we now have proof that plague is plague not just over centuries but over millennia. It's the same organism causing disease in this setting, even going back that far. And the people doing this work, they never stop. They then get a high-coverage Yersinia pestis genome. They looked at unique substitutions. They looked at they found some false positives in the previous genome, and they corrected themselves. And then go forward a bit further, and now we've got multiple Yersinia pestis genomes from across Western Europe, showing slight variations between them, showing that there was slight diversification during that first pandemic. So it's just remarkable how we're moving forward with this. But as I say, they don't stop. They keep producing papers. So you move forward a couple of years further, and here we have Johannes Krause's group looking at genomes, Yersinia pestis genomes, from the Bronze Age. So although the earliest evidence of Yersinia pestis has been identified in the late Neolithic early Bronze Age, these strains lack key components for flea adaptation. So it was kind of unclear, what does this mean? Is this plague as we know it, or is it some kind of predecessor? And they show here that they managed to get ancient Yersinia pestis genomes from the late Bronze Age period, and they show that there are clear distinctions between the new strains that they've got and the lineage that has already been described that causes the flea-borne infections, and it suggests that but they did show that there was ability for the strains that they'd reconstructed to be involved in flea-mediated transmission, and suggested that actually plague as we know it did actually evolve a lot earlier. And a more recent publication from Rasmussen and Villerslev shows similar kind of things, but they're now looking back into the Neolithic period and into the Stone Age, and showing that this is going back nearly 5,000 years, and what they're suggesting is that the spread of plague
coincided with the first large settlements in the middle of the Eurasian subcontinent, continent, and mass, and that the organisms spread through trade routes, through the use of the wheel, through animal traction, and so forth. And what they showed was that the different populations where they were able to show that plague was present, the human populations were actually genetically distinct. So it wasn't that a group of humans moved around Eurasia carrying plague with them as part of their kind of endemic infection of that population, but instead the infection was clearly spreading between different populations, even that far ago, long ago. So that's a remarkable story. It's not a story that's over yet. I'm sure if we come back next year, a couple of years, 5 years from now, there'll be even more of these plague genomes and even older ones, and we will be able to see every step in the evolution of this infection over time. But now I'm going to move on to a second case study, which is that of leprosy. So leprosy is a chronic infectious disease of skin and peripheral nerves, caused, at least classically caused, by an organism called Mycobacterium leprae. Mycobacterium leprae belongs in the record books because it is actually the first bacterium that's identified as causing a specific disease in humans. So this Norwegian guy here, Armand Hansen, back in 1873, was able to visualize the organism down the microscope and show that it was reliably associated with patients that had what was clinically deemed to be leprosy. So leprosy is sometimes called Hansen's disease in his honour. And what happens with this disease is that it destroys the nerves, and this leads to anaesthesia, peripheral anaesthesia in the hands, the feet, and so forth. And what happens is that people end up cutting themselves. They get, you tread on a stone or a needle or a nail or something, and it makes a lesion in your leg. You don't know anything about it. The thing festers, causes tissue damage. It's not the actual organism itself that's causing the damage. It's the fact that when you have no feeling at all in your hands or your feet, that you just keep damaging yourself as you go about your day-to-day business. I mean, there's one story from one of the leprosy colonies where a guy was trying to explain that you have to look after yourselves. You do a visual inspection of your fingers every day to check that you haven't hurt yourself, and if you do get a cut, then you dress it and whatever. But there was one individual whose fingers were slowly getting smaller, even though there didn't appear to be any reason for it. And then it turned out what they discovered was that at night a rat was coming into the bedroom and eating the man's fingers while he was still alive, and the guy just didn't know that's what's going on because he had no feeling whatsoever. So this is a truly terrible disease. And here's just some images. This is an old Norwegian man with leprosy from over 100 years ago. So Norway was one of the last countries in Europe to have very appreciable numbers of leprosy patients. Here's an individual from South Asia, and you can see here the characteristic lesions you get where you end up losing your fingers. In fact, the whole reason I became a microbiologist is I read a book, it's kind of a variant of the Lord of the Rings, that featured a man with leprosy going into another world, being cured of his leprosy and so forth, and that stimulated my interest in leprosy and in infection generally. One of the great things, we almost be too negative, is that leprosy has become curable in the last few decades with this directly observed multi-drug treatment. You can go from looking like that one on the right to on the left there. It is actually curable. And the number of people suffering from leprosy in the world, when I was your age, it was around 8 million. I think we're now down to the hundreds of thousands of individuals with active leprosy. So this is a problem we are solving. Now, the first genome sequence of the leprosy bacillus was a major undertaking around the turn of the millennium. This is the paper here, Stuart Cole. It has been an enduring influence in this field, the leader in the field really of studying the leprosy bacillus and its genome evolution. And what we discovered with this very first genome sequence, and it was one of the early genome sequences in the first half dozen or dozen genome sequences to come out, what they discovered was that this genome sequence showed an extreme case of reductive evolution. So what I said earlier about what happened with Yersinia pestis, where it got into a new niche, got very specialised, and then could throw away most of its genome. Mycobacterium leprae had done that, but even more dramatically. So less than half of its genome contains functional genes. But there are lots of pseudogenes where you can see by looking at the sequences that there was a gene that worked there once. It's similar to what you see in, say, Mycobacterium tuberculosis, but it's no longer functional. And this is because presumably the ancestor was a free-living organism, lived in soil and had to fight for existence and struggle, get lots of nutrients and do all sorts of things. But all that Mycobacterium leprae does is live in humans and live inside Schwann cells and scavenge an existence in this very particular niche. Another interesting point was that the leprosy bacillus, Mycobacterium leprae, at that time was believed to only exist naturally in humans. And there was no environmental reservoir. The only way it got around was human-to-human spread. But they discovered that if you tried to grow it in laboratory animals, it wouldn't grow in normal mice. Well, it grows very neatly in the footpad of the mouse. But if you use nude mice, skid mice that have no immune system, then you can grow leprosy. But the best organism is a 9-banded armadillo. I'm not quite sure how anyone stumbled on the fact that you could infect the 9-banded armadillo with leprosy, but it turns out you can, and it grows in the 9-banded armadillo to huge amounts of bacterium, and it's what they used here to get the genome of the leprosy bacillus. They had to actually grow the leprosy bacillus in the armadillo and effectively put the armadillo in, kill it, and then put it in a blender and harvest all those leprosy bacilli to get enough. Because the other thing I forgot to say is that you can't grow this organism in the lab. Because it's lost so much of its genome, it's not capable of living in the lab and growing on normal culture media. It will only grow in host tissues. So a few years later, we started getting multiple genomes. And so what they found was that I met Stuart Cole just before this paper came out. And he was saying he was having great difficulty finding any variation in the organism at all when he took leprosy samples from patients scattered across the world, from South America, from Oceania, from Asia. Really, it was very hard to find any variation at all. But in the end, they did manage to get it down to found a few rare single nucleotide polymorphisms, and they told a story. As you always do with these things, they made a story to say that the disease probably originated in Eastern Africa or the Near East and then spread with human migrations, and that Europeans or North Africans introduced leprosy into West Africa and the Americas within the past 500 years. That's what they said at the time. As I was discussing all this over lunch with some of my colleagues, if you get to a certain level in this field, you can keep publishing papers in Nature every few years where you do the same work, you just do it better with the latest tools and a bit more. So they come up a bit later on, and they've now actually managed to get some complete genome sequences of various strains from around the world, from the Thailand, the United States, and this strain from India. And they showed then that they got differences in only 215 polymorphic sites, mainly SNPs. And they discovered that there were 16 SNP subtypes throughout the world. Now, obviously, when you do these kind of studies and you get loads of genome data, you can't just say, well, I've got loads of genome data, I've not been good. You have to kind of tell a story. So, again, their kind of just so story was that there's a strong geographical association that reflects migration patterns of early humans from trade routes, and they blame the spread of leprosy on the Silk Road linking Europe to China, contributing to the spread. Now, that was with modern strains. Then a few years later, they came up, particularly once Johannes Krause got involved, many of the same individuals were involved in getting medieval leprosy genomes. So what they did, and I didn't talk about this with the pestis genome, but one of the tricks that you can use is you can use a capture-based approach. So you can use DNA sequences from the modern genome to make probes that you can pull those sequences out of your samples and massively enrich for the pathogen sequences from your sample. So the trouble is, when you're looking at any kind of bit of bone from a long time ago, it will have huge amounts of bacteria, contemporary bacteria, that colonize that bone after death. And trying to work out what looks like the pathogen in the midst of all that noise, the genomic noise, is quite hard. So what they did here, they used this trick where they actually made probes, they fished it out, and they compared the ancient, what they're calling ancient, I mean they're medieval, Mycobacterium leprae genomes with these with 11 modern strains, and they showed, again, remarkable genomic conservation. So again, they put to bed the idea that maybe leprosy back then and leprosy today are different things, because the same arguments apply as with plague. You know, we call it leprosy, but is it really the same thing? I think there's still arguments about what they call leprosy in the Bible, whether that is leprosy as we know it today. But basically, medieval leprosy appears to be the same as modern leprosy. The genome is very, very conserved. It's the same kind of organism, and this speaks to the question of what was it that led there to be so much leprosy in Europe back then in medieval times, and so little now. Obviously, one answer is that it was a different kind of strain. There was some kind of super clone of leprosy that swept through medieval Europe. It was more virulent, more transmissible, or whatever, than what we have now. But it doesn't appear that that is the case. It appears that basically it's the same organism in both contexts, and therefore the only explanation of differences in the way humans live in human society, human health, and so forth. So, remarkable European origin for leprosy in the Americas, and a genotype in medieval Europe that's now commonly associated with the Middle East. And so, this was a remarkable step forward. We basically had medieval Mycobacterium leprae genome sequences, and here are the samples that they analysed. A sample from Winchester, two samples, one a tooth and one a skull, from Denmark. Two different samples, and then some from Sweden as well. And you can see they're all from the mid-range, the Middle Ages there, 11th and 13th century. And when they drew phylogenetic trees, what they're able to show is that you've identified these 5 different branches. Branch 0, the outlier, branch 1, 2, 3, 4, and they show that the samples that have come from medieval Europe sat within these 2 branches, 2 and 3 there. And they nested, so branch 3 is defined like a medieval one, branch 2 is defined like a medieval one. Branch 3, they could show that those samples were similar to one from the USA, so presumably carried to the USA by European migrants. Now, I said earlier that leprosy is caused by Mycobacterium leprae, and that's what we thought up until about 10, 12 years ago. And then there was this remarkable discovery made where I think it was in Mexico, in Mexico, they found that there was a patient with leprosy that was clinically fitted all the criteria for leprosy, but was caused by a different organism that was only distantly related to Mycobacterium leprae. So this new organism is called Mycobacterium lepromatosis. And when its genome sequence was announced, they showed that it had undergone a similar kind of evolution to Mycobacterium leprae in that large amounts of the genome had been lost or under a process of decay, that there are lots of pseudogenes, kind of faint echoes of genes that used to be functional there. But this, I think they have a common ancestor that was about 1 million years ago, I think. So it completely set, most recent common ancestor 13.9 million years ago. So it's a remarkable example of convergent evolution, where two lineages of mycobacteria that were presumably free-living decided to jump into humans and maybe other animals and cause this particular disease, adopt this particular lifestyle. Now, as you get old in science, your ability to be surprised goes down. You probably find everything in the world fascinating because you never come across it before. But as you get old, you do start to sort of think, oh yes, and like I've said, yeah, another leprosy genome, yeah, oh, we've got medieval, okay, yeah, that's interesting. But, and this is looking at the lepromatosis and leprosy, showing you how different they are from each other. But this is one of those papers where I just, WTF, you fall off your chair. I mean, it's just what the? I mean, this is unbelievable. So the dogma was that leprosy only lives in humans. There's no environmental reservoir. And then it turns out, just a few years ago, that they could actually find leprosy bacilli in red squirrels in the British Isles. Just totally unbelievable, just amazing. And not only that, not only did they find it, they did genome sequencing. And what they showed here, so Mycobacterium lepromatosis has only been described a few years before. It turns out that it's there in squirrels in England, Ireland, and in Scotland. And that the original classical Mycobacterium leprae is also present in squirrels from Brownsea Island in England. So this is just amazing. And they said that there was disease, so it does appear to be a case associated with disease in these animals, but also found in some healthy animals. And the British and Irish Mycobacterium lepromatosis strains show that they are part of the same species, but they're diverged 27,000 years ago. Whereas when they looked at the leprosy strains from squirrels, they found that they're actually quite closely related to the strains that were found from medieval England, circulating in medieval England. So this is just showing the phylogenies here of the squirrel strains in Mycobacterium lepromatosis. The squirrel population does appear to be quite different from the human one, and whether there was some event tens of thousands of years ago where it jumped from squirrels into humans or vice versa, or whether it was independently acquired from a third host, we don't know. But when we look at what we see in Mycobacterium leprae, we see clear evidence from the phylogenies that all these squirrel genomes are nested within this branch here that includes these two medieval leprosy genomes, one from Winchester, one from Denmark, and this one that we mentioned earlier from the USA. So it's clear that there has been spread between humans and squirrels. And looking at the fact that squirrels are nested in one branch of the diversity we see in humans, probably squirrels called it from humans. What I haven't said is there's a similar study that shows that 9-banded armadillos in southern parts of the United States actually are now carrying leprosy in the wild, and they caught it from humans as well. And there are some naturally infected non-human primates now that have also been shown to carry leprosy. So the dogmas that you grow up with, what it says in the textbook, most of the time it stays true, but sometimes it turns out not to be true after all. And this was one of those. And this is the most recent paper looking at this, where they've looked at a range now of genomes, a new set of ancient leprosy genomes in Europe, and they found that there is this much more diversity than they expected. And so their previous stories about all the silk routes or the slave trade, whatever, they have to kind of pedal backwards and say, well, actually, it looks as if there was huge amounts of diversity in Europe very early on, and that maybe Europe is where this disease started. It's interesting that they can keep getting papers, high-profile papers, but they can contradict themselves as time moves on and show that their earlier papers are not quite true. So again, come back in another few years, we'll have more of these leprosy genomes, we'll have even better insights into how things go. Right, the last case study is on tuberculosis. So this is caused by another mycobacterium, Mycobacterium tuberculosis, discovered by Robert Koch very shortly after M. leprae was described. And this is a disease of the lungs primarily. You get fever, you get chronic cough, you get weight loss, night sweats, hemoptysis. You do really sometimes also get infection of other organs, extrapulmonary tuberculosis as well. And the genome sequence of Mycobacterium tuberculosis was one of the earliest genomes to be sequenced from bacteria. And this is a phylogeny that was drawn up in 2002 by Roland Brosch and his colleagues, where they actually plotted the ancestry of modern TB strains from this TB complex, as they called it, where a bunch of related strains that infect various hosts. Some of them infect the so-called M. bovis is found in cattle, but it's also found in seals and goats and oryxes. And this is the story that was told back then. But when the M. bovis genome came out the following year, they had to change their tune. So the idea was that we caught TB from cattle when we domesticated them. It turns out that, in fact, the M. bovis genome is a shrunken version of the M. tuberculosis genome, and therefore it looks as if M. bovis was derived from M. tuberculosis rather than the other way around, or that we, in fact, gave the infection to the cattle rather than we acquiring it from them. So again, this is I hope you've already, in what you've learned about evolution up till now, you know about the role of just so stories in this game, where people try and make narratives and whatever. It turned out this was another example where they had to overturn their original thinking to say that actually it looks as if it spread from humans to cattle rather than the other way around. Now, I got involved in this area. It's an area where we have done some research a few years ago, and basically we entered into a program of research that had sort of been started just after the turn of the millennium, where this guy here, Mark Spiegelman, who's a remarkable individual, he's a Holocaust survivor. He's got a cousin who lives in New York, who's written a book called Mouse, which is a graphic novelisation of the Holocaust. And Mark is there as one of the children moaning about the fact that he's hungry. After the war, he left Europe, went to live in Australia. Then, around the time of the Yom Kippur War, he decided he'd become an Israeli. And he's also got a UK passport. And he's a surgeon, but at the age of 50, he decided he was bored of just being a surgeon. So he went off and did an archaeology degree and became duly qualified as an archaeologist and as a surgeon. And he got involved with Ilko Pak, who's shown in this picture here, a Hungarian scientist, paleoanthropologist, in a study where they looked for, they discovered, in fact, Mycobacterium tuberculosis in 200-year-old human mummies. And At the time we got involved, there was evidence from PCR that this organism was there. There was even some microscopy evidence suggests the organism was there in samples. Here is Mark using an endoscope to actually take lung tissue from one of these mummies. But it was unclear whether this was true or not. The same dismissive stuff, well, it's all just PCR contamination, was in the air. And we got approached by Helen Donoghue saying, would you be interested in looking at some of these samples, see if you get genomes out of it? And I said, well, we're not going to do any PCR or anything like that. What we're going to do is just use this approach of shotgun metagenomics, where we just extract the DNA from the samples and we sequence it and we find what we find. And it means that we can find stuff in there that we don't expect to be there. We don't have to make a probe knowing what we're looking for. We'll just see what happens. Now this is one of those studies where she approached me and I said to the postdoc, look, this is high risk project, probably not going to give us anything, but when you're not busy, have a go and see what happens. We anticipated there'd be lots of human DNA in the samples that we were given, and we knew that there would be post-mortem contamination with all sorts of other bacteria as well. And so I kind of thought if we got one sequence alone that would just be unequivocally TB in the whole library of sequences, that would be a hit. But I kind of got ahead of myself here, because let me just tell you the story of these samples in more detail. So the samples came from this place called Vax in Hungary, which is in the middle of the country, on the border of the Danube, on the banks of the Danube. And in fact, one of my former colleagues, Judy Brewer, her father was also a Holocaust survivor. He actually fled from Hungary just at that point, swam across the Danube just near there to escape. But anyway, what happened was in 1994, a crypt was discovered in this church, a Dominican church, and it contained 242 bodies. Now the crypt had been sealed up and not looked at, people didn't even know it was there for over 150 years, but it was used for just this time period from about 1731 to 1838. And anyone who was kind of middle class and had enough money to pay to be in the crypt, and the various clerics associated with the church, were put into that crypt. And for reasons that are still not clear, many of the bodies that were put in that crypt became naturally mummified. So instead of all the flesh just rotting away and just leaving bones, much of the soft tissues remained. And this gave us a remarkable opportunity to look at these samples. And our first efforts centred on this young woman, Terezia Hausmann, who died on Boxing Day in 1797 at the age of 28. Now, as part of the previous studies around the turn of the millennium, they'd done chest x-rays on these patients, these mummies, and she didn't have any lesions that suggested she had TB in the chest x-ray, but she did appear what we call cachectic, which is a highly wasted individual, very, very, very thin. And that is consistent with TB. And the previous analyses that Helen Donoghue and Mark Spiegelman and so forth had done on the sample had confirmed that it looked like TB, and it suggested very good preservation of TB DNA. So here's a picture of her mummy. Here's a picture of her death record here to show you that she died here on the 26th of December 1797. So this was an interesting facet of this work, was that we did have good documentary evidence about who these people were and when they died, in addition to having their bodies. Often you find bodies, in the plague pit, you have no idea who these people are, and you only have very, very rough estimates of the time that they died, whereas here we knew for certain what happened there. My postdoc, Jackie Chan, she did some metagenomics. She got 5.5 million reads, and surprisingly, hardly any of them aligned against the human genome. But 8% of the reads did align against the Mycobacterium tuberculosis reference genome, which gave us about 32-fold coverage of this genome. And again, this is one of those knock you off your chair kind of moments, because that's more or less what you would get if you just grew TB in the lab and then extracted DNA and sequenced it. It's just absolutely remarkable coverage. Now, the first thing is, wow, that's amazing. Second thing is, this is probably wrong, isn't it? It's an artifact, can't be true. But we thought about it. We'd never grown TB in our lab. We never performed any PCRs in the lab. And anyway, we had deep and even coverage of the genome. So we said, well, this must be real then. We've got a 200-year-old TB genome, which is truly remarkable. One of the bioinformaticians in the group started analysing the data, and he got quite annoyed with it because he couldn't get proper phylogenies that worked and looked reliable. And then he realised that what was happening was in these pipelines, he was excluding any parts of the genome where there wasn't at least 80% of the reads agreeing. And when he removed that criteria and looked at in detail himself, he found that in many parts of the genome there was evidence of 2 different alleles, or 2 different variants at a particular position in the genome. And so what this meant was that there was a mixture of 2 different strains of Mycobacterium tuberculosis, 2 different genotypes in this individual. And there had been some rather more rudimentary attempts to type the strains in these samples previously using an approach called spoligotyping, and that had suggested that there were 2 different genotypes circulating in the Houseman family. So in Terezia Houseman's mother, I think, had a separate genotype as well. And what Martin Sargent, the postdoc, did was he looked around the genome, and basically the coverage you can see there is pretty much even. So that suggests we're not just looking at contamination from an environmental mycobacteria, which might have a little bit of similarity to TB in certain parts of the genome, other parts which show no similarity. There's a big spike there. I think that's because that's the 16S region, the ribosomal operon, which is very conserved. Otherwise, the coverage is pretty even. But then he looked at certain parts of the genome, and compared to the reference strain, suddenly it just drops off. So what that means is that in this 200-year-old genome, that region of the genome has been lost in the lineage. But what he also found when he looked carefully was that there were some regions of the genome where the coverage dropped by half. So instead of being 30x, which was the kind of basal background coverage, it dropped down to about 15x. And that, again, was evidence that there was a mixture of 2 strains, one of which had a deletion in that part of the genome, and one of which did not. So that was very interesting. So we wrote all that up. We got a paper in the letter in the New England Journal of Medicine. We drew our own little just so story tree of showing how the type strain H37RB, these mummy strains, and one of them too, and a lineage that was circulating in Germany at the time differed from each other. These deltas are the regions of the genome that got deleted at various stages in the evolution as time went on. Now, shortly after we did that, we were the first to do ancient TB genomes. But this paper appeared from, guess who? Johannes Krause, working with people at the Sanger Centre, Sanger Institute in Cambridge. What they had found was they got 1,000-year-old samples from pre-Columbian mummies from Peru, these skeletons from Peru. And what they managed to get when they analysed their genomes was that instead of having M. tuberculosis in the strict sense of the strains that usually infect humans, what they found was that there was this strain called Mycobacterium pinnipedi, which is nowadays generally associated with seals and sea lions. But they found that these pre-Columbian individuals had actually been infected with this pinnipedi. So this is the skeletal samples, this is the site where they got it in Peru. And they actually used our dating. They used our Hungarian mummies as a calibration point to actually date the most recent common ancestor of this TB complex, which included Pinnipedi isolates, suggesting that TB perhaps arose about 5,000, just over 5,000 years ago. And this is their phylogenetic analysis, and you can see here the variation of the various lineages that are found within this Mycobacterium tuberculosis complex, and what they discovered is this Pinnipedi. Ours over there was the genomes we found within this L4 clade over there, and this is how they actually showed the number of variants and SNPs that define their lineage. We went we went further with our samples. We realised that actually it wasn't just one sample that was going to give us a good signal. We actually looked at 14 genome sequences representing 12 different genotypes from these Hungarian mummies, and we were able to. We'd have probably got a Nature paper if they hadn't published theirs. They undercut us by getting their paper out, so we got to Nature Communications, and we were able to get a much broader range of results. So we had the Terretia Hausmann genomes, but we also got it from these other individuals, 7 other individuals, 2 of them with that not named, but the others named. And we found that all of these genomes belong to this particular lineage 4, or Euro-American lineage. We got a variety of depth of coverage here. In some cases, just less than 1x. But in some cases, absolutely astonishing. When we went back and did Terretia Hausmann again, we were getting over 200-fold coverage of the genomes there. And in one other of these samples, we were getting 187x coverage of a genome. And this is still unexplained as to why is there so much TB DNA in these samples. It must be that I think that the TB continued growing after the individuals died and scavenged their tissues and grew to a much higher biomass than you'd expect. Having said that, nobody's gone in and sampled a horrible tuberculous lung in the worst case nowadays and seen how much TB DNA you get out of it, but it is a remarkable finding. And we were able to draw our own big phylogenetic tree here, where we took all of the known sequences at the time from this lineage 4, and we could show our various 200-year-old genomes and place them on this lineage here. And we thought that basically the origins of this lineage went back to probably the late Roman period, around 400 in the Common Era. And this was consistent with what the people at Sanger had said, that maybe the TB complex originated about 6,000 years ago. It certainly conflicts with some PCR evidence of Neolithic TB, and the question's still open as to whether when TB actually moved into humans and when were the first human cases. But again, another point that I should say is that these TB strains that we analysed from 200 years ago, they are very, very closely related and nested within the diversity we see in modern TB lineages. So there was nothing different about the TB strains in Europe 200 years ago than the ones we see circulating today. There was no superbug TB that ripped through Europe back then and has gone away. It's clearly there was a difference in society, in health, in the population density, general levels of immunity and so forth that accounts for these differences. Now, one of the interesting findings that we had, I'm almost finished now, is that many of these individuals have more than one strain. So what we saw with Terretia Hausmann, where we saw those 2 different strains, turned out to be quite common among these 200-year-old samples. And again, this is one of those things where you thought, what? Really? Is that common? And this probably is a difference between what we see now and what we saw then, because nowadays, if you look to see how many times someone with TB had multiple strains, the presumption is they just get infected once in their life, they get one strain. But again, that's a presumption, it's almost a dogma. And the way in which we do TB diagnosis is we grow the organism in the lab and then we pick a colony and we follow up that one colony and do work on it. We don't pick 20 colonies and see whether 3 of those 20 are different or 5 of those 20 are different from the others. But where people have done that, it seems that, particularly in KwaZulu-Natal, which is one of the highest areas in terms of prevalence nowadays, up to 1 in 5 cases appear to have mixed infection. And so what we saw back then from metagenomics, it actually recontextualizes what we're seeing today as well. And when you think about it, if you're going to be coughed on by someone who's got TB because you're living in this very highly pressured environment, everyone living very close together, why is that only going to happen once in your life? You know, someone could cough on you a week later, a month later, a year later, with a different strain of TB. And so why would you not get infected more than once? It kind of turns things on their head. Now, this work actually attracted the attention of Bill Gates. That gave me bragging rights with my children. Because, you know, most of the time what you do, it's all rather arcane. But the fact that Bill Gates took an interest in it was pretty cool. But of course, there is an issue here. There's no new cure for TB, but actually the approach we'd used hadn't been used on contemporary samples. And obviously, if we're looking at 200-year-old diagnosis, it's a bit late to make any interventions. And so I said, why don't we try this on contemporary samples? And we were the first to actually try that, where we, the PhD student working for me in Gambia, got sputum samples and just extracted DNA and sequenced it, and we were able to detect TB genomes in those sputum samples using the same kind of approach without having to culture the organism first. And so that was an interesting example where the past and studies on dead people kind of illuminate what we can do in today's world. Okay, I've pretty much finished now. I've just acknowledged the various people that have done some of the work that